Yay! Hi, welcome back. I am Anahita, and there is Maya and Maria. Thanks for attending our third workshop about high throughput phenotyping with for root. But before starting, as you know, we do a very brief presentation about PhenomeForce, who we are, and what we do. And then we will move with the speakers of today that will have a very exciting workshop. So with these, I give the word to Maya.
Thank you, Narita. Again, welcome back to this 2022 season of plant phenotyping. This year is dedicated, we are focused on root phenotyping. And this PhenomeForce was born a couple of years ago as a fantastic initiative to share knowledge and tools and current resources on plant phenotyping. Initially, it was created by Anarita and Felipe. And this year, Maria, myself, and Gurlen, that is under hurricane and cannot join Gurlen at the moment. So we have joined also this initiative. And with that said, well, you can learn more about us in our GitHub website. There's a further description and links to all the resources. You can also follow us on Twitter for all the updates and the upcoming workshops and past ones. And all the recordings of all the workshops, past, present, and the ones coming, will be forever available on YouTube, in our YouTube channel. So you can go there and find how to contact all the speakers and all the resources they share. Any question, you can always contact us by email, and we're also on Slack if you would like to join the community.
Yeah, with that said, I would like to welcome our speakers. Today we're going to have two speakers, Peter from the University of Georgia here in the United States, and Dr. Khwan from Mahidol University from Thailand. And the floor is all yours. Also, they are going to be talking about phenotyping root hairs using microscopy, images, and dirt mew.
Well, thank you, Khwan and Peter, for accepting our invitation to present today. So we want to remind everybody that if you have a question for the speakers, you can write them in the chat, and we will ask the speakers to answer your question as soon as they can. And with these, so I guess Peter will be speaking first, and so please, guys, good luck with your workshop, and we will be here to support you anytime you need.
All right, thank you. I guess I'm live now. So yes, I will be presenting how to use dirt mew today to phenotype root hairs. And this project basically started at the very beginning of my PhD. I'm now at the end. And we were thinking this would be just like a small piece, but it became very big and took up most of my PhD, actually. Actually, I met with Khwan or Patompong Saengwilai. I think it was the first month of my PhD, and that's where we started it. And we thought we would create this high-throughput phenotyping for root hairs. Because it's very tedious to do it by hand, it's not very accurate, and we just wanted to improve this part. And with that, I would actually like to hand over to Khwan. He's going to give an introduction why root hairs are important and how they take the images and do the experiments. And later on, I will explain more about dirt mew and later on go through the whole pipeline to process the data.
Okay, thank you so much, Peter. So I'm going to tell you, I'm not going to spend too much time on this topic because I believe the exciting part would be the demonstrations of how to use Peter's tool. But I'm just going to tell you a little bit about root hairs. If you are not familiar with root hairs, it is just this one single cell and that extend into the soil. So the whole structure is just one single cell and it's elongate into the soil space, and it's usually developed at the differentiation zone, just right after the elongation zone in the root system. Now, there are different types of root hair formations, and most of you who read textbook, are probably familiar with Arabidopsis root hair, which is usually the type 3. In this type, you have the distinct differentiation between the cell called trichoblast, which is later forming root hairs, and a trichoblast, which is not forming the root hairs. And there's the really distinct pattern where you see the cells that forming root hairs and not forming root hair alternate, among each others. But other plant species like rice, maize, and other crop species, usually in type 1 or type 2, where any cells can form root hairs, or there is a symmetrical cell divisions that's starting, and smaller cells start forming root hairs and bigger cells not forming root hairs. So there are many type of root hairs, that just to let you know. There are some great reference out there. Marsac is one of the one. If you want to study about development and evolution of root hairs, this is what I kind of recommend you guys to look at. Now, functions of root hairs has been quite intensively studied, mostly in Arabidopsis, but lately in many other crop species as well. We know that root hairs help for soil anchoring. It's help with seedling establishment. So it is quite a good trait when you look for root system or root traits that will help with seedlings adaptations. But what it's known for is it's help increase the root surface area, and it's estimated that up to 80% of nutrient acquisitions come from root hairs. Now, in many crop species, we have shown that in the infertile environment, such as in low phosphorus, low potassium, or even under drought, having a little bit longer root hair seems to be beneficial trait. And that is, simply because when the root is growing through the soil, if the rate of uptake of nutrients is fast, faster than the diffusion efficient of nutrients moving from the soil to the root, it's create this gray area called depletion zone around the root system. So by extending a few hundred microns, to the side of the root, it's help pretty much increase the absorption of nutrients under low soil fertility. And in our study, our recent study have shown that not just in phosphorus and potassium, low phosphorus and potassium soil, but in the case of nitrogen, root hairs also seems to be beneficial as well. So it seems like a good trait, and I would say it is really good indeed. As a root biologist, I have been working with root traits for probably more than 15 years now. And if I were to choose one root traits for breeding program, I would recommend root hairs. And that is because root hair it has low metabolic cost, meaning that, if you pick plants or segment of roots with root hair or without root hairs, the metabolic cost of those root segments are pretty much, the same. And this is done in Arabidopsis in the control environment. Now, another cool thing about root hairs is that it's developed independently from other root traits, which open this opportunity for breeders to put different traits together with root hairs. And another thing is, because it has been intensively studied, a lot of study has shown that it works. And by saying that it works, I don't just mean, in the greenhouse and in the experimental field. It does work. And I want to suggest or recommend you guys to take a look at these papers by James and Jonathan Lynch. This is a beautiful story putting together breeding targeting root traits, and particularly incorporating root hairs into the traits that allow common bean to grow in a low phosphorus environment, and which results in the release of 3 different common bean varieties and helping people in Mozambique. So this is quite a beautiful piece of work. If you want to look for a story or inspirations of working on root phenotyping or any phenotyping, this would be a good paper to look for, to read. Now, how to study root hairs? I think it's, can start very simple. You start with the seeds, and with the system. It could be any kind of system, but from my experience, when we study roots, I tell my students like it's not just any system. You need to understand also that a lot of assumptions about these root phenotypes come from the behavior of nutrients and resources in the soil. So, for example, if you want to study root hairs for low phosphorus conditions and you screen root hairs in low phosphorus nutrients, such as in hydroponic system, it may not work, because in hydroponic system, you probably already know it doesn't have that diffusion limitation that they have in the soil. So when I want to study root hairs, I usually go for soil medium or soilless medium or soil mixture that allow the plant and soil or medium interactions that would kind of like imitate the condition in the field. And this is the pictures of my actual root hairs experiment that we did and published for nitrogen study. We are using the system called mesocosm, and the medium is simply sand, perlite, vermiculite. And the thing that you can notice is that the soil, the column is pretty long. It is around 1.5 meter, 1.6 meter long. And we want to use the long tube to study roots because we want to create this gradient of nutrients, like what happened in the field when you fertilize plants in the topsoil and then you irrigate. There is the gradient of nitrogen and phosphorus and other and water in the system. And by using the system that is a little bit more realistic, your results can then be applicable to the field as well. So this is kind of the system that we used. A lot of people ask questions about, okay, root hair seems to be really fragile. It is only one cells. How do we extract it from the soil? My answer to that question is just try it. Just try and see. You will be surprised how tough the root hairs are, and it's actually pretty tough. However, if you want to study root hairs from plants that you collect from the field or a little bit older plants, you also have to realize that when plants get a little bit older, they shed the epidermal tissue. So sometimes you don't see any root hairs at all if you collect root from the field, and that is probably because the root portion that you collect is pretty old. So you need to look for the younger portion of the roots. Now, after you extract the root from the soil, simply by kind of like gently checking it and rinse it with water, you then go through the root imaging, which I will show in more detail in a little bit, and then the image analysis part and statistical analysis, just like any other trade you do. So this is how we do it in the lab. This is a simple setup. We usually cut the roots into 1 to 2 centimeter segment, although this is not required. I have worked with other species. This is rice, by the way. When I work with other species like corn or common beans, sometimes we just put the whole root system in the color stain. The stain that we use is toluene blue, 0.1%. We can use. I have seen people use trypan blue as well. So it depends on your preference, and you just have to try, to see what's worked for you. So we dip the roots into the color stain for 1 minute or less. Again, depend on the concentration, depending on your setup, and then we rinse it twice with the distilled water before we took it for imaging. In our setup, we use microscope for imaging, but I have used stereo microscope as well. So again, it depends on how big the segment. And something like that. So this is the kind of setup that we have. We can then take picture using the iPad. Okay. Back in the day, I even tried to use the camera mounting on the microscope and take picture that way. But now we are a little bit more high tech now, and this is the kind of image we usually get. I mean, it's good because you can see it's good contrast. But time to time we get really bad photos, which I think I have some to show you as well. So from this image, what we have to do, or what we usually do, is to go through a bunch of image analysis. A lot of us use ImageJ. If you are not familiar with it, it's a great tools for any simple image analysis. You can write your own plugin, a macro or something like that. But for root hair length and density, this is very simple. So you choose your scale and you do the set scale. You know 500 micron equal to maybe 222 pixels. You set the scale and then you start measuring your root hairs. So for measuring root hairs, usually people pick 5 to 10 representative hair. The more the merrier, but you take the more time. And so it's 5 to 10. That's what we have been using. But also in one plant, we pick 3 representative roots, and within the root segment, we select multiple places. So all of this will lead into why I have to talk to Peter about developing the program, because it's all laborious and tedious. But this is before the program. So after we do that, we select the representative root hair, and you can actually see if the root hair is damaged or if it's intact. Okay, so just like this one, you can select the healthy one, and you can just measure. That is for root hair length. For root hair density, there are several ways that I have seen people do it, but the one that I find the most consistent when we measured it manually is to look at the linear density, which means that you draw a line. Usually this is 1 millimeter length, and then we manually count the number of root hair that is in the same plane, which is in the right focus, and then we can calculate the density this way. And with those methods, it's people have been using it and report typical root hair response. And usually when you deplete them from phosphorus and potassium, you see the increase in length, increase in density. But when you reduce the nitrogen, people reported that it's reduced in length and density. And what I have seen is that this is not always the case. We look at different cultivar, different varieties of the same species, and they seem to respond very differently. Like in this picture, you see that this one cultivar of corn, having these long root hairs in the control, when we deplete them with low potassium, instead of getting longer, it's really actually reduced. But when this variety, a little bit shorter, but when we deplete them of potassium, it's become longer. So the response is depends on the genotype. On top of that, there are different root classes, in the root system. Rice have even different type of lateral roots, compared to maize, which have crown roots, seminal roots, and primary roots. Common bean having basal roots and a bunch of lateral roots. So to make it more fun, the root hair response of different root classes are different. So the location, the root class you collect can influence your outcome, your result. So, and oftentimes you come up with this one, you get this kind of images, and then you asking yourself, which one are we going to pick, for length and density. And that is why I start talking to Alex and Peter about, how can we make this phenotyping technique, phenotyping ways of root hairs more accurate and more consistent, because, to select 10 root hairs from this is actually very subjective. So I think that is what I would like to talk about in my part, and I will hand it over to Peter, who is helping me looking at this and developing this program.
So I did prepare a couple of information that you might need for the workshop, actually. So you might actually do it now. The information should be also in the description in the YouTube channel. I just wanted to put.
Before starting, I think there is a question for Khwan. I
Mhm.
don't know if you answer. So it's what do you think about root hair phenotyping in semi-hydroponic systems?
In semi-hydroponic system, you mean kind of like with substrate and irrigating nutrients? Is that correct? I mean, I have seen people screening root hair for breeding program in many systems, using many systems, some of which, just straight germination plate and just look for the shortest and the longest root hair. So I think it depends on your question actually. But if you want to study like physiological response and using hydroponics or even semi-hydroponic system, um, I think it depends whether the the behavior of your resources, is kind of like what it is in the soil. For example, I mentioned phosphorus because it is what known for root hairs. And when you study using hydroponic or even semi-hydroponic system with low phosphorus, you don't have that diffusion limitation that they have in the field. So there are a lot of studies in rice already using comparing the response of root hairs in different systems like hydroponics, pots, and the field, and they actually come out differently. So I don't know if that answers your questions, but it's up to you. It's up to the resources that you want to study, something like that. Yes, the good questions, really good questions.
Thank you, Khwan. So then we will leave the words to Peter now.
All right, thank you. So yeah, you can basically maybe if you I would suggest actually to go through this after the workshop, and it's probably better if you just follow me. I will explain all the detailed steps, so it might be difficult to both watch and do it yourself in parallel. But yes, anyway, so there are a couple of reasons to measure the root hairs automatically. And Khwan already mentioned a couple of them, but like in summary, it's more objective. You can get more information out of it. It's faster. You're able to process more images per root, so you can even like increase your sample size, and it's more fun as well. So actually counting all these root hairs manually on ImageJ, from what I've heard, is that people do not like that. There have been previous studies of softwares to measure root hairs, and one is here by Vincent, where they used machine learning to classify root hairs, and these images were taken in the soil. They actually did a decent job considering that it's in the soil. Another one is by Gouchard, where they created this profile of root hair length. So this is also like actually lower resolution image than we use. But yes, they are also able to determine basically, I would say it's still a proxy for the length. And what we wanted to do basically is to identify all individual root hairs, because that would result in more tangible traits like root hair density and the distribution of root hair length. And when we started this a couple of years ago on rice, it was actually quite easy because rice has like a generally lower density and shorter root hairs. And we are also able to resolve these intersecting parts. And you can see that not all root hairs there were segmented, but that was also at the very beginning. And then later on, we started with the more complicated images like maize, and we saw the method is not really working. The results literally looked like spaghetti. And it also ran forever. So some of the images didn't even like finish after processing them for a couple of months. So we had to make a lot of improvements to and reduce the computational complexity of the algorithm to be able to measure these high density and long root hairs. And as Khwan mentioned already earlier, like which root hairs in this image are representative. You can see a lot of long root hairs, but then there are also a lot of shorter root hairs, and some root hairs, like on the other side at the top, they are a bit longer than at the bottom. So yeah, that might make sense to actually measure all of them to get a representative sample or measurement. So we came up with this pipeline basically for DirtMu. So when we take the image, we use Elastic, which is a free software that you can download to create a training set. And we selected, so we label or annotate the pixels that belong to the root hair, which is shown here in red, and the root in blue, and the background here shown in green. And then we do this for a couple of images, and then using Elastic, we can, in the batch process, classify all the images that we have. And this will give us all the pixels that belong to root hairs. And then eventually, what DirtMu does really is it extracts the skeleton, and based on the skeleton, we extract the root hairs. And you can see here that some of them intersect, and the main point of DirtMu is resolving these intersections. And the way this works, I have this little demonstration here, is basically we fit these segments like randomly, and they are not necessarily belonging to one root hair, so they can, like here, for example, you can see that one that curves. And they are like random, just strips of splines that are fitted to the root hairs. And then we use an optimization algorithm called simulated annealing to fill the image and resolve intersections and create complete root hair. So we have a cost function here, which is basically reducing the curvature, reduce the number of remaining root hairs, and also minimize the distance of the root hairs to the root. So this part here would be, for example, far away from the root. So if I run this, you will see that after so this one will run for almost 20 million iterations. And we throw in like new lines or take out new lines. And at each iteration, it gets a little bit better. And as you can see now, we slowly get to the point where we have all of the root hairs. And at this point, only a few changes made, so it looks pretty good. You see that not all root hairs are perfect, and there are also like these small outliers, but we can remove these, especially the small ones are easy to remove. And what we get as a result is basically this image, which all the individual root hairs extracted. Some are missing. We can also identify the main root and the lateral roots, so that we know if the root hairs belong to the main root, whether that's the primary root or whatever, or the lateral root. Most of the time, we're interested in the root hairs that grow in the main root, and we want to remove the ones that are growing on the lateral root. And we're also able to detect the edge of the root here. So this is shown in green, and in blue the bottom edge. And in this way, we can determine the density. And yes, so what we get really is the distribution of root hair length, as well as the root hair density and total at the top edge, the bottom edge. We can also find the area that has the highest root hair density, and we get individual root hair measurements like length. We can also measure the curvature and actually also the diameter, but the diameter is very noisy just because of the low resolution. And a couple more. We have a couple of measurements for curvature, so the total curvature, also the part where the curvature is the highest. So that's curvature. We normally don't want to measure it, but we had for Ankita's research, for example, it was useful. She had found these hooked hairs. All right, so we also validated our results. So in this case, we had 15 images where we measured, or one of Khwan's student actually measured all the root hairs manually, and we compared this to the automatic measurements. So the automatic measurements are shown in blue, and in orange the manual measurements. And you can see that across all the 15 images, they look very similar. And these were images. We took the images with short root hairs and long root hairs, high densities and low densities. So we have a representative case. And we use common bean, maize, and rice in this case. And in summary, so when we look at the correlation between the traits like mean length, between the manual and the automatic measurements, we found really good correlations, high correlations. The automatic measurements were of the root hairs were a bit shorter. But if you look at the maximum length or the longest root hairs, we saw like a perfect one-to-one relationship almost. We also recently looked into the standard deviation of the length, and that correlated as well between the automatic and manual measurements. And then for the number of root hairs, it's a bit lower than for the length measurements, and that's mainly caused by the images that have like a very high density, where the root hairs are so close to each other that it's nearly impossible to distinguish them. And this is one of the cases where it becomes really difficult to classify it and also then to extract it with DIRT-mu. And of course, if you have your own data set and you use DIRT-mu to extract root hairs, you can also create like a small validation set if you're unsure if the results are correct. So you just take, I don't know, maybe 10 or 15 images to make sure that everything works fine. All right, so we did a quick sanity check to distinguish between species by root hair length. So this is common bean, where the root hair length is around 200 to 250 micrometers, and rice was much shorter, and maize had the longest root hairs. And this was from a large data set. We also had different treatments. So we had the control and reduced nitrogen, phosphorus, and potassium. And we had about 3 to 4 replicates for each treatment and 5 to 7 images for each replicate taken along the route, which resulted in more than 700 images. And we then automatically extracted almost 140,000 root hairs. And that took, in this case, around 4 hours. But I had access to a lot of processes, so that's why it was possible to do it this fast. And the bottleneck was really a couple of images that took a long time. So especially the maize images. So what you get really is the distribution of root hair length. That's like the additional information that you get. So rather than just taking the average of, let's say, 5 root hairs, you get the complete distribution of root hair length. And I don't want to go into the details of this, just basically to represent that you get much more data. And it also makes it more reliable if you get the length of all the root hairs. So one thing we notice also is that there is variation along the route. So this is just one plant and 5 images taken along the route. And in 2 of the images, the root hairs were actually much longer, which we can also see here in the histogram. And then for the other 3 images, the root hairs were shorter. So that's why I think it's also really important to increase the sample size, have at least 3 images at different locations taken along the route. And if you can automate this, it's just very easy. So it does not cost you much more time to take the images, and it takes you no additional time to actually process it because there's no manual work involved in that. So we compared our measurements also to the standard manual measurements that Khwan mentioned earlier, where you only measure 5 representative root hairs, and the density was measured along a representative area. And what we saw for maize is that the manual measurements were always much longer than the automatic measurements. And this was a trend we saw in our validation set as well, but it wasn't as extreme in this case. And this basically suggests that the manual measurements were not very representative, so that the student actually took the longest root hairs. And root hair density actually correlated still quite well. There's a lower correlation than we had, in some cases at least, than we did in our validation set. All right, so let's move on to the actual workshop. So I want to give a short summary of what we're going to do. So the first step that we will do is basically classify the microscopy images using Elastic. And Elastic is a software that was developed by Anna Pietrzyk's lab. It's free, you can download it. I've been using it for many years now. It's very user-friendly. You don't need any knowledge about machine learning. And in this way, we will get these classified images. So we have the 3 classes for background, root hair, and the root. Then we will upload the classified images to PlantIt. And PlantIt is basically the website that was developed by our lab, more specifically by Wes Bonelli. He's been working on that for a couple of for the past 2 years. And yeah, so we will upload the classified images to PlantIt, and then on PlantIt we will run DIRT-mu, and that will result in the measurements of the root hairs, and eventually also download the results from PlantIt. So yeah, once again, here's the information you will need if you want to follow along. There are a couple of other prerequisites, which is taking good images. Khwan mentioned that earlier. And we tried DIRT-mu in a couple of different images, so just standard RGB images as well as grayscale. dark field images where the background was actually black, and also enhanced depth of field where you stack multiple images at different focal planes. That also works. You should have sufficient magnification, so the pixel size should be smaller than 2 micrometers. So for the big data set that I just showed, the pixel size was between 0.67 micrometers for the rice images, where you are able to zoom in more because the root hairs are shorter, and 1.72 for the maize images, which were zoomed out a bit more. However, if you want to, of course, compare different measurements, so let's say within the rice data sets, you should always use the same magnification. That was one issue that we had, so we could not just create one training set for the whole data set. We had to split it by magnification. The images should have the same exposure, so make sure that none of the images is like much darker than another. You should have a high contrast such that you can see the root hairs very clearly. Root hairs should be in the focus, of course. And in DiRT-mu, we prefer that the orientation is horizontal. And this enables basically to extract the top edge as well as the bottom edge. The images should be relatively clean, so there shouldn't be like too much dirt, soil, or any fungi in there. Especially fungi are difficult because the hyphae, they look a bit similar to root hairs. They are actually a bit thinner, but it's difficult for the classification as well as DiRT-mu to distinguish root hairs and fungi. Yeah, also make sure that the scale is in a good position and not covering too much of the root hairs. And DiRT-mu actually also doesn't like if there are like any lateral roots on the edge of the image. So like here, sometimes it like struggles to determine the main edge because that is based on the diameter of the root, and the highest diameter in this case will be here. So yeah, make sure there's always like some space like here at the bottom at the edge of the image. All right, so the first step will be pixel classification with Elastic. And this is the software that we will be using. So we'll just create a project for pixel classification, input the data. We will select the features that we want to use, and then we will spend quite a time on the training. And normally I would suggest to use between 10 to 20 images for the training, and label each one of them, and then iterate through the whole process until you're satisfied. And then we will go through the prediction export and batch processing. But for this workshop, I will only use 3 images for the training, just because we do not have enough time to go through so many images. And later on, we will upload the classified images to Cyverse or directly to Planted, actually. So there are 2 ways to do that. You can upload it to the data sets on Planted, or to your data sets on Cyverse. It does not really matter. I will also show how to do both. And finally, we will start the DiRT-mu workflow on Planted. So we will log into Planted using our Cyverse accounts and go to workflows, select DiRT-mu, and then submit a job there. We do have to select a couple of parameters on DiRT-mu. So the first one is the number of levels. That's important for the optimization process, but you can just leave it to 200. You can go a bit higher if you want. So I think the maximum, set the maximum value to 1000, but then it will, the whole process will run longer. And then we can also set weights for some parameters, but you can also leave these to 1. So we tested these, and actually, yeah, leaving them as 1 gives good results. And I might actually remove all of these in the future just to not confuse anybody. What is important though is to specify the IDs for your root background and root hair labels that you used in Elastic. So these IDs should match the labels that you used in Elastic. And you should also calculate the pixel size in micrometers. Or if you set it to equal to 1, the results will be basically in pixels and not in micrometers. And yeah, calculating the pixel size is quite easy. So if you have the scale on your image, you can use ImageJ, for example, to measure how many pixels long the scale is. and then you just divide. In this case, we had 200 micrometers, so you divide 200 micrometers by 298 pixels, because this distance from here to here is 298 pixels, and it's also shown here. And this will give us a pixel size of 0.67 micrometers. And eventually we will download the data from Plantate or Cyverse. All right. And finally, I'll also explain you the files that you will get. So one will be just showing the image with the extracted root hairs, and the most two important files that you will need for data analysis. And one of them ends with root hairs, and the other one with summary.csv. I think at this point I also want to make some acknowledgements before I go into the actual workshop. So I want to thank Alex and Wes for their support in developing this, as well as Khwan and his students who gave a lot of the data that we worked with. And I didn't show any of Mark's data, but he also gave us a huge data set that we've been working with. And that's everything for the slides that I have. And I will move on.
Hey, Peter, if you want to make a little pause, there are a few questions that we could already address. Would you like that?
Yes, sure.
So I think the first one was from Ashish. He was asking if you could input resolution segmented image to dirt. He's having some troubles.
Sorry, what was the question?
Could we input resolution segmented image to dirt?
Yes, I think that should work. So basically, any segmented image should work. You can also, I never tried classification with ImageJ, for example, but there's also, it's called a Weka classifier. So technically that should work. As long as the output file with the segmented parts is in TIFF format.
Excellent. And the other question is from Jody. Can this workflow be changed to measure other features such as root length?
Yes, I'm actually planning to do that. Yeah.
Excellent. And I believe that's it for now. So thank you so much so far, and please continue.
All right. So I talked a bit about Elastic, and it's this free software, so you can download it from elastic.org. If you go to download, make sure if you on a Mac you use the newest beta version, I think. And let me start this. So all right, so when you start Elastic, you will basically create a pixel classification project. That's what we used. Another one that could be useful is pixel classification plus object classification, and this would allow you to remove small segments that might be like floating around, or maybe they are like too short root hairs. But I will stick to the pixel classification for today.
Hi, Peter. Can you increase a little bit the font or the size?
I don't think I can actually.
Okay. Well, okay. That's
in this image, but yeah, you click here. It says pixel classification. So I will save this here. I will rename it to PhenomeForce. And you end up with this screen where you can add new images. And I already prepared a couple of sample images. And one way you can add images is just by dragging them in there. And I have 5 images. I will only use 3 for training. So let's take this one. I'll add this one and this one. And later on, we will actually, in a batch process, process all of these images. And here you can see basically just an overview of the images with a nickname, the location where it's saved. For TIFF images, we don't have an internal path, and then also like the shape, so the resolution, and we have 3 bands for red, green, and blue. The second step then, or maybe one thing I should mention is that for some images, Elastic did not accept them, even like TIFF images, but when I resaved them in image.j as TIFF, it worked. So I'm not sure exactly what went wrong, but so you might select not the usual technical issues, but usually there's a workaround for that. So the next step then is to select the features. So I usually select all the features, and what that means is that for the first one, it is going to play a Gaussian smoothing to each of the bands at different sizes. There are also edge detection filters and texture filters. And I have had good experience just selecting all of them. If you like, you can also add like Gaussian smoothing or edge detection filters at a higher scale. So this might be useful if you have a very high magnification where the root hairs are like have a very big diameter in terms of pixels. But in this case, I don't need them. And I will show a couple of examples how that looks like. So if I, for example, take the red channel with a very low smoothing, it will look basically the same, but if we apply a lot of smoothing, it will remove some of the noise. This might take a second. So just bear with me. Oh, there we go. So yeah, now you can see that everything is a bit blurry. And similarly, if you look at the edge detection filters, it will give you some information about the edges between, for example, between the root as well and the background, as well as the root hairs and the background. And this always takes a while. The fan of my laptop is running really loud now, so that's why I'm using the headphones. So here's an example of that. Let me check if I'll show you another one. Yeah, this one, for example, is really nice. You can really clearly see here the edge between the root and the background. And for root hairs, so this is really helpful to actually identify the root hairs. That's why I use all of the features and at all scales. All right, so then we move on to the training, and we will need 3 labels. So I will add a new label here. And I will go quickly through the navigation. So as you saw already earlier, you can here maximize the images, or you can go to the full resolution. Here you can add labels. I usually relabel or rename these just to make it easier for me to remember what is what. So I just do this to root and 3 for background. And Elastic will only save the numbers. So these labels are already assigned IDs, like 1, 2, and 3. And even if I would write here, I don't know, 5, the ID would still be 1. So you should basically stick to the correct order. And here we have the navigation cursor and a brush tool to annotate our pixels. And we also have a brush to remove anything you want to. You can also change the size of the brush, what is it called? The eraser, as well as the brush. You can change the size, but I always stick to 1 for the brush. And later on, we can also click the live update, and this will give us an intermediate solution. All right, so let's start by adding a couple of labels to the root. And what I normally do is I just select the couple of very clear root hairs. I select a couple of pixels there. You don't need to label the whole root here. So just going like in the center of the root here is enough. You can also zoom in more. Zooming works, I haven't mentioned that. If you press Control and then use the wheel on your mouse, you can zoom in and out. And if you just press the wheel on your mouse, you can drag the images. All right, so let me select a couple of other root hairs. And for the first iteration, I really just take the, let's say, the good root hairs. If there's like something a bit blurry, I would not do that in the beginning. Maybe also here. And then I can go to the root labels and select the pixels that belong to the root. And a couple of areas that I know are already tricky are like here, for example, at the lateral root. And can already go to the background. So I can just select a couple of pixels here. And it's going to be important to also select pixels that are close to the root or to the root hairs. So I usually go like this. And also here. And if I click Live Update now, it's going to show me the probability of these pixels. So I can see that the root has the highest probability for root indeed, and the root hairs, the root hairs, etc. You can also click on segmentation, and this will give you the classified pixels. And one very helpful tool is this uncertainty here. So this will basically show you all the pixels where the classifier, which is a random forest classifier, where it's not very sure yet which pixel it is. And this will improve as we add more labels. All right. It's also good to actually turn off the live update while you're selecting pixels. Otherwise, your computer will run a lot and it will always constantly update. And at this stage, I would really already move to the next image and do the same here. So select root hairs. Maybe go across as well. Yeah, maybe here we have these two root hairs that are intersecting. I'm going to select this. I think it also goes further here. I hope you can see that there's actually a root here. I can see it on my screen, but it's already a bit fainter than the rest. And then here I can also select a couple of pixels that belong to the background. I'm going to be very careful still. And the same for the root. And here you can see that the transition from the root to the background is also a bit blurry. So I would be a bit careful with labeling this space here. Normally, I just leave it as it is and for now, and might improve it later. So I want to label all this. These lateral roots always cause troubles or areas like this. So I'm going to label this as well. Because you can see that it's clearly root, but also have these blurry lateral roots. So I can label this very carefully, and I'm not exactly sure where the root starts and where the background starts. So maybe I can get a bit closer. And we can improve that later as well. So I can click the live update again, just to show you how it looks like. And the live update will update all the parts that you can see. So if you actually zoom in it will only update the area that you can see on your screen. And you can see the whole image shot's gonna classify all the pixels. Yeah, so we can see a couple of problem areas that we might have to look at later already. So I'll still leave this as it is. Maybe it's not reacting. All right. Maybe we can already add a couple of background labels here. And I will move on to the next image and pretty much repeat what I just did. I might do this a bit better. So it looks like there's a root here. Here, but I'm not going to label it. So I've really focused on the really good root hairs at the moment. And here we have this weird transition again. So at this point, I would be a bit careful with this. Also have to be careful always that you actually place the correct labels. That's why I write the names here, so I don't accidentally label the background as root. That has happened. And then you struggle to find where you made the error. All right, so I'm gonna add a couple of more labels here. As well as the background between the root hairs. Maybe find another area. So these are obviously root hairs. All right, so at this point, I added labels in all of my 3 images, and I only selected like the areas where I was very sure that it is indeed a root hair, background, or root. And at this point, I would go back to the first image. Run the live update and see what needs to be improved. And this looks quite good to me, but I also see there are a couple of problem areas here at these lateral roots, so we will need to add labels there. And I always like to add the uncertainty as well. And yes, you can also see that here these spots are very uncertain. It looked like it was not labeled or not classified correctly. All right, so turn off the live update. And I can work a bit on this. Yeah, it's tricky. So I would actually do it like this. Maybe again, click the live update now and see if it improved. And I can also click on segmentation. So that should actually work. also want to update this, see if we can improve this. I think it's still updating the classification. So it's the tricky thing with this live update. It's not the fastest, especially if you move around and add labels. Also have this bad habit of actually turning on the probability, but not really seeing the raw image. So it's probably better to turn it off. Well, I'm not very happy with this, but I will leave it for now and maybe focus on another area, like here. Then you can label this as root, and this as background. I really want to focus a bit more on the root hairs. It looks very good, actually. We check the uncertainty. So a couple of these root hairs are a bit out of focus, so you can probably barely see this. And it's always a question like, are you going to use it for your measurements or not? Here's one that's also a bit blurry. I would actually still label this. I would not label this one as root hair. And I would also not label it as background because it might have an adverse effect on the actual root hairs. I think there might be two root hairs here. If you're not sure, then just leave it. That's what I always do. Yeah, it also somehow sees a root hair here, which I do not see. So I might actually label this as root. And there will always be some noise, of course, and it's never going to be perfect. Sometimes you get these halos around the root here, so I like to add a few background labels here. Just be careful not to get too close to the root here. I think this will also be a problematic area. So everything is very uncertain. Yes. So I would probably pick the best root hairs here. Wow, this got much better. Better than I expected. Might try to put some background in between here, but might be a bit risky. I would probably leave it as it is. I think if I add more labels here, some other areas might get worse, and Can you look at this here? Looks like there's a root here. See the whole image, see if there are like any obvious errors. I would also not add any labels to the scale. I would just leave it as it is. And because this is like floating in the image, it's going to be excluded anyways from the results. So this will be just an outlier. All right, so we have a couple of things here, and here at the top right. So I'm going to look into. Yes, so this might not be very important, but I'm just going to leave this as it is. It's not getting better. Okay. It's also not the most important part. In this case, at least, because this lateral root is also just floating, and it will be excluded. All right, let me check why. So move on, iterate to the second image. I think I have to work a bit in this area, in this area. Yeah, this is difficult. This is background, background here, and here. I'm not sure what to do here. All right, that's quite a bit of work that we need to do here. So I think I can label this as background. You also have to be careful when you have, for example, the uncertainty view on and add labels. So I use this a lot, but then you have to double check that you didn't label anything incorrectly. And maybe this is also one of these areas that you just have to leave as it is. There seems to be like another lateral root very far in the background, so that's why this area is a bit darker. So it will probably never get perfect. And we'll have the same issue here. Yeah. I would focus on the root hairs that are really looking like root hairs for now. This is also a very faint root here. I can see it on my screen. I'm not sure if you will be able to see it. I try to add a lot of intersections because all that dirt mu does later is resolving these intersections, and if they are not, if they are a bit if the classification is a bit noisy, it will be more difficult to resolve these intersections. And even add a couple of pixels here. All right. Now let me check. This metal root looks good. So there's this root here. I'm thinking whether I should label it as root here or not. It's definitely a root here, but it looks a bit different than the other root hairs. So if I label it, it might have a bad effect on. There's like one root hair that looks weird, and you label it, and it might have on the other 100 root hairs that look actually correct. But I think for this workshop, I'm just going to try it. I can see that these root hairs here are in focus here at the base of the root hair, but when they move away from the root, they tend to be a bit out of focus. The question then is whether you should add labels here or not. And I'll also double check. They are barely visible. But if you want to get the whole length of the root hairs, I would still add these labels here. And it seems like the classifier is able to pick this up, even though it's very faint. All right, I think this looks also good enough. Maybe we can add a couple more labels here. All right, and now we'll move on to the last image. I'm going to spend a few minutes on this. I think the results looked promising. I might have to work a bit here and here. Okay, this area looks good. Let's wait for it to update. And yeah, so normally if you have like 10 to 20 images, yeah, you will be busy for a couple of hours. So I'm just doing this now in half an hour with 3 images. But you can expect to work on this. Or let's say I would work on this for, let's say, 4 hours. But I did this already many times, and it might take you a bit more time to do this. So you might spend the whole day. Oh, I should have turned off the live update. Yeah, this is one of these, I think it's a root hair here. Could be also fungus. It looks a bit thinner. I'm not going to label it, neither as background nor as root hair. I'm pretty sure it's a root hair, though. There's a little bit of space between these two root hairs. Check if this improves. Oh, this one also looks a bit weird. Maybe I'll just leave it. I hope this is going to improve. Oh, I did get a bit better. And I think I'll just check the whole image and then move on to the export. I could spend more time on this, definitely, improve it. And another thing you could do is go back to the first image and double check, make more improvements. Make sure that the amount of labels that you add to the background root hairs and root is about the same. So make sure you don't add, let's say, 1,000 labels to the root hair and only 10 to the root. But you will see that it wouldn't give you good results. Anyways. All right, so I'm going to turn off the live update. I can then go to prediction export. And you'll see your data sets that I used for training. And as a side note, I think it would have been actually smart to move the image into a separate folder so that you know which images you used for training. You forgot to do that. And here you can select what you want to export, whether it's the probabilities, simple segmentation, uncertainty, features, or labels. And we really just want simple segmentation. So I could export each of these now, and they would be exported to the folder where the original file is. But you can also make changes to all the settings. So you convert to a different data type. You can renormalize the min-max, and I will show that later, whether that is useful. You can change the data type. So Elastic really likes to work with HDF5, but DirectMu likes TIFF. And we also want to change the directory and the way we name our data. So I'm going to select the create a new folder for classes, save it. And yeah, this is also a bit tricky here. So I'm not sure if you'll be able to see this, but it's these are like curly braces, and it says dataset dir. in it, and it should be nickname in there, which will then select the data set name, basically, Pietzanulog minus P1 minus 3, and so on for each of the other ones. And then after the curly braces, I like to write classes. You don't have to do it, but that's how I do it. And now you can see here the updated folder and the file name. So it's basically Pietzanulog and then underscore classes TIFF. All right. So if I export one of these now, this will take about 20 seconds. So when I export this image as it is now, it will look like a black image because all of the pixel values are either 1, 2, or 3. So these are the label numbers that we had. Yeah, so it looks pretty black. It's not very convenient if you want to double check if the segmentation is good or not. So one way to do that, basically, to fix this, is to renormalize from our labels, which were between 1 and 3, and we renormalize it to 0 and 255. And then if I export this, you'll be actually able to see the classes or the segmentation. And currently on DirectNew, it doesn't accept this, unfortunately. So I will change it. I think it's like it would streamline the process a bit. I just want to show you that you can renormalize the labels. It's still processing. All right. Yes, and now you can see basically the image renormalized. So that's just like one piece of information that I think is quite valuable. But yeah, for DirectNew, at the moment, still turn off renormalization, and I promise that tomorrow or so I will actually change that so that it can have values between 0 and 255. And that means basically the lowest one will be 0, and then in this middle one will be 127, and 255 for the background. All right. So now that we set everything here in our settings, we can go to the batch process. And I will just take all of the 5 images that we had. So we used 3 of these for training. I will include them in the batch process and classify all of them. And yep. Oh no, that's not good. Maybe. All right, I don't know what happened there. But it started processing now. So now it's a good time to ask questions, because we have to wait a minute or two.
Can you hear me?
Yes.
Yeah, thank you, man. That's very cool. Hi, everyone over there. Felipe here. Actually, I have a question. Do you have any trained data that we can use as a start point for example, I want to start my experiment right now, and I want to, start from something already trained. Do you have these?
So yes, we do have trained data, but I think you really need to create a new training set for each data set. And the reason is basically there are like so many different types of images that you can use. So I work mainly with RGB images, but if you have, for example, grayscale images, you will definitely need to create a new training set. And the same applies for if you use a different magnification. or if you would use, for example, a different stain. So all of these images are stained with toluidine blue, but if you don't have that, it would make I don't think you could use like one training set to classify a different data set.
Okay, so it's very data dependent, right?
Yes, yeah.
Okay.
I think maybe if the training data set it's really huge, it might work, but I'm not sure. And I know that Elastic also implemented neural networks, still in the beta phase. So maybe that would work, but I have not tried it yet. So I only use this normal pixel classifier, which uses random forests.
Okay. So you do have a question here. Let's see if Maria de Oliveira can make it. Maria?
Yeah. Here, I think it's this one. Can you download the configuration of the classification to reuse it?
I don't think so. No. I think that's related. I think it's really specific for each project or data set. Yeah.
Okay, and I also have another question here from one of the participants, and he asks, like, how can we ensure that the data of length and density obtained by this algorithm is accurate and true?
Yeah. So yeah, I did show. So we did a bit of validation already on it, and it was actually, yeah, we saw that when we measured all of the root hairs manually, we actually got very good correlations. If you don't trust these, you can always manually measure a couple of your images manually, and then see if that correlates well with the automatic measurements.
Good.
But it might not give the same results. So I mean, it's which measurements are better, the automatic ones or the manual ones? So even if you measure all of the root hairs manually, you probably do it a bit different than the algorithm does it. So I think it's never going to be like perfect.
All right. We have another question here from Jonathan. Let's see. If we have a set of 1,000 images, will it be enough to make the training with 20 and then upload everything?
So if the images look very homogeneous, I think that should work. If like some images are darker or have a different slightly different hue, you might need to add more images, I think. So what you can also do is just add like 50 images or more, and just select fewer pixels fewer pixels per image for your training set. I think if the training sets on Elastic become like very huge, it tends to run very slow as well. So I would not overdo it with a training size. That's why I always use the pixel size of 1 for the brush. Basically here,
Thank you.
All right. Okay. So let me continue. All right. So we do have all of the classified images here, and now we can move on to upload them to Planted. And for Planted, you will need a Cyverse account, so you can sign up for free. If you pick the basic account, you get 5 GB of storage space. And then you can basically log in with your Cyverse account into Planted. And I might make this a bit bigger. No, I think this is not going to work. Yeah, so it's just planted.cyverse.org. You can log in. This goes automatically. And let me just give a little tour of Planted. So you can get additional information here, what Planted is. There's also documentation. So this might be useful if you want to check how to use it exactly. I think if you go to Quick Start, and then to submitting tasks. I think this will be the most useful information for this workshop. But I will always also go through this step by step. Right. And you can check the status, the stats, as well as the status of the platform. Oops, okay, don't log in again. Okay, and then on the left side, you see workflows. You will select Dirt New. There are also other workflows for counting stomata, for example. There's also the original Dirt platform or software, as well as the Dirt 3D reconstruction, and a few others, or one other. Then you can go to data sets, and here is where you will find the same folder that is on Cyverse, basically. And you can add a new folder by clicking here. So I'm just gonna create one, PhenomeForce. It will take a second. There it is. So if I click this little button here, let me create another directory for the classes that we just got from Elastic. And yet another for results. All right, there it is. Let me check what happened here. I clicked the wrong button. All right, so if you click here, if you're here at the results and you click this, you can upload files. And I'm going to pick all the classes and then press upload. And then you have all your results on Cyverse. I'm going to want to go through a couple of other things. So yeah, I also have projects here, but I did not start any yet. Also the agents, so that's the high performance cluster where your data will be processed. And right now, Sublow 2 is available for everybody. So you should select this one. And I think you will actually only see this one. I don't think you will see this one and this one. These are actually closed and you need special access. If you go to tasks, you see an overview of the tasks that are running, the ones that are completed, or if you have any scheduled. And you can also check users. And yeah, so we have the data sets here. I just want to quickly show you that if you go to the discovery environment on Cyverse, so that is de.cyverse.org, you will basically see the same data sets. And you can also upload your data from here or download it from here. And if I refresh, yeah, you'll see that I have PhenomeForce here and the two folders that I just created on Planted. So it's basically the same. I think this one has a couple more features, like if you want to rename it. I don't think that's possible on Planted at the moment, metadata or details. All right, let me check. Okay, let's go back to Planted. And we will start a workflow, a Dirt New workflow. There's some information here that's not important. So there's mainly the Docker. So Planted actually pulls the code from GitHub and updates the Docker container automatically every time I change something on GitHub. And then that container is basically used to run or to process the images. And there's also a link to the NAPPN article from the beginning of this year. I'm still working on a research article here. But yes, you can then click submit. And here's where you can change a couple of parameters. Like you will be assigned a random ID for this task. you're free to rename this, but it has to be a unique ID. And you can also specify the time. So maybe you want to think you need more time to process all of the images if you have a large data set. And here's where you select your input. So actually I have a backup PhenomeForce folder created, but I will select the data that I just processed with Elastic. So go to PhenomeForce here and select classes like this. Yes. So now it says PhenomeForce classes, and I will do the same for the output. PhenomeForce, and I'll pick the results. So that's where my classified images are, and I want to save the results in this folder. Then I can select the agent here. I think you only see subload 2. And I tested it yesterday. It was running a bit slow. That's why I will use subload 2 batch. It was running a bit faster yesterday. You can create tags. So, for example, PhenomeForce. And these also rights images. So I could add this. And you can change the parameters that I mentioned earlier. So we're going to stick to 200 for the levels, and we're not going to change the weights for the optimization. And then these IDs for the root, background, and root here, these should match the ones on Elastic. So it's good to actually double check. So we had 1 for root here. That's correct. 2 for the root. All right, and 3 for the background. And you can change the pixel size, which I need to change here to 0.67. All right, and you cannot, no. So you have to press the minus and plus buttons. And then you just start the project, and I hope it's going to work. So this will bring you to a new screen, to tasks, basically here, where it will show you the process of your task. Now it's had it prepared the environment, and soon it should start processing the images. And this will, so in this case, for these 5 images, I think it will take at least 10 minutes. Oh no, I don't know what happened. Did I forget anything? Let me check. We uploaded. I did this yesterday, and it worked yesterday. Just going to make sure that we have. Does say no?
Peter,
Yes.
Jonathan is saying that you have uploaded the classes image on results folder.
Oh, did I? Oh, okay. Yeah, that's what I thought. I didn't upload them, but I just uploaded them to the wrong folder. Yes, thank you. And
Thank you.
I will actually, yes, I will delete them from here. I could also just swap the folders in the task parameters. Or I can go to, let me double check. This is classes. Okay, let's do this correct this time. And upload. Okay. Let's try again. So the settings should be all the same still. Okay, just double checking. My text disappeared, but that's fine. We don't need them. I just want to make sure that the parameters are correct. And let's try again. And yeah, like I mentioned, this will take at least 10 minutes. So we can wait a bit, or I can actually download the files from the task that I ran yesterday. Just want to make sure that this does not crash this time. So prepare the environment. I'm going to wait a few more seconds. Yeah, and I'll schedule the job. So I hope it will be running and give us results in 10 minutes. But I can also as well, because I ran a backup yesterday, I think this will be faster. So you can go to your folder. It's basically the same structure. Go to results. And that's where you can see all the results. So all the images, the CSV files. And you can also just download the zip file, which contains all of these files. It just makes it easier. PhenomeForce. Let's create a new folder for results. All right, so here's the zip folder. Actually unpack it. Might move around a couple of files. I think it's like nested in a couple of folders now. But yeah, so the results for this image will look like this. So this is just an image to illustrate you if it worked well. And you can see that that identified the lateral roots, and the root hairs that are growing at the lateral roots are shown in red. It actually looks like did a decent job. It did not take this as root hair because it's sort of it grows from here and then moves into the lateral root, so it wouldn't be possible to measure the length. So that's why I label it as outlier later on. All right. So for the same image, you also get a file with the outliers, and here you can see that this was, even though it looks quite neat, it would actually classify it as outlier. And all of these little parts here, so these are good, well, even here, yeah, some funny parts, or like really short segments. So these would be all labeled automatically as outliers. And the most important file here is probably here, the one that says root hairs at the end. And that's going to give you a spreadsheet with a length for each individual root hair, as well as the different curvatures. So this is the total curvature integrated over the length, and this is in radians, the average curvature, median, minimum, maximum. We also have the diameter, also measured in micrometers, and you can see that most of them are around like 10 to 15. So there's a variation. There should be really the same. So we have a median as well for that, min and a maximum. And then also very important, so I think the length in micrometers as well as the root class are the most important columns here. And this one will tell you whether it's a lateral root or whether it's the main root. And if it's a main root, it would also tell you if it's at the top edge of the root or if you go all the way down at the bottom. So these are the measurements for the individual root hairs. There's also a summary file, which you can use to calculate the density and a couple of other things. So for example, there's the area that was labeled as background, as root, and as root hair. Then the length of the root or the edge of the root at the bottom, at the top, as well as the total edge. The total length of all the inliers, so these are the actual root hairs, and also outliers. So we'll see that the total length of the inliers is much higher than the total length of the outliers. Also, the ratio that we were able to resolve, it's about 9% of the total length that we have in the image was actually not resolved. And then also the root hair count at the bottom, the maximum root hair root hair count, that's in a window size of 10. So it's basically an area on the root that has the highest density within like a 1 millimeter width. Average root hair count based also on this moving window, as well as the minimum standard deviation for that, and then the root hair count at the top, the total root hair count. We also have the root diameter, and yeah, basically the window size, but that's always 1000. So this is a lot of information. Sorry about that. So the most important things that you will need most likely is the total root hair count, and then the total length of the edge. And this way you can calculate the density. So it's just the number of root hairs divided by the length. Yeah, and you get this for all of the 5 images that we processed, as well as some other output from Planted. This is basically just output that DirtMu gives. It's not really relevant. Just in case there are like any errors, you could check for that. And I think that's all I have. I'm gonna quickly check here how my task is doing. All right, so this is still running. Okay. So we started this 7 minutes ago. I think it will still run for a couple of minutes. So I think it's not worth waiting for this. And it will be the same, pretty much the same result that I just showed. Okay. Let me know if you have any questions.
Yeah, I think Gerling, she has some questions for you. Hold on a second. Gerling, you are muted.
Hi, my name is Caroline, and I'm also part of the PhenomeForce. Great presentation. Thank you so much. I have a couple of questions from the registrants. One is, this protocol can be used to separate rules in 3-dimensional images or not?
No. So at the moment, it really just works on 2D images. So I'm planning to adapt this to work on actually complete roots, but not to identify root hairs on like complete roots. So it will actually look into the root architecture. And that's going to work on 3D as well. But we're not planning to do this for 3D sets of root hairs. I think there are not really many. You would need a CT scanner to do this, and I think there are not many around.
Great, thank you. And also, I think Felipe already talked about, like, is there any training data set for a new crop when you start a new crop?
No, so I worked with rice, maize, and common bean. But yes, you definitely need to create your own training set. And it really does not take so much time. So maybe like at some point in the future when there's like a bigger community and people have a lot of data sets that are maybe very similar, that would be possible to share the training data. But I think it would be ideal to actually create a training set for your individual data set, just because there are so many differences in microscopes and experimental setups. So all of the images generally look different. And I've seen so many different images. And I don't think that one training set would work on the other.
Great, thank you. Yeah, those were all I had. And if the attendees have any question, go ahead, Maria.
Yeah, I actually found another question from one of the attendants, and it is like, how clean do the root hairs need to be because it is a very time-consuming step, right?
Mhm.
So he would like to know if it has to be very clean or not.
Yes, I would say fairly clean. If there are a couple of like spots, it's fine. But I think one of the early data sets that I worked with, it had a lot of fungi on it, and it was really, really difficult. And the results, or the quality of the results, was not as good. I think it's possible, but definitely the quality of the results was suffering.
Yeah, I can add into that because I'm in the first, the first portion of the process, which is the growing the plant and obtain the root hairs. So you can actually see the root hairs under the microscope, and you can go through the root hairs. In the slides that I show, my student cut off the root hairs to like a small section because we are using microscope for taking the images. But what I have done in the past is to look at stain the whole root system, put it in a plate, and scan using the stereo microscope, scan through the whole roots and find the most representative part. Now, when we collect the samples, we usually look for the middle part of the root axis, because if you go toward the base, that's where there is the flux of the hormone of the plants. The root hairs seem to be too long. When you go toward the tips, that's where the root hair not forming yet. So usually the middle part is where we aiming for. And of course, if you grow in the soil, the media is dirty. Obviously, you have some fungus. So just make sure at that part that we get the clean root hair.
All right. Thank you for clarifying.
There's another question actually.
Yes.
Is there any methodology how to scan roots images without having seeing the negative effect of the circular nature of roots? Maybe a better water bath based bath?
Mm, yeah, it's very difficult, and I think it's also very complicated. So the image is always like a 2D projection of a three-dimensional object, and normally the root hairs will grow in all directions. So I think as soon as you put it on the slide, it will be also a bit flattened. But besides that, you still have the 2D projection of the microscope. So it's very difficult, and I think it does not matter so much. I think there's a little bit of effect. And what you could also do is use EDF images. So if you take like a stack of images that focus on different parts of the root, you might take away some of that, but it's still a 2D projection. And I don't know exactly, I don't know what is better for measurements, if you use this enhanced depth of field, or if you have like a very narrow slide that is in focus of the root hairs. I'm not sure what's better for getting like the measurements that are most relatable to the 3D length. But yeah, that's a good question.
That seems
very difficult.
Yeah, I mean, it's a very good question. I mean, I have seen people using like growing plant in a gel medium and doing the 3D, measurements, trying to get everything surrounding the root hairs. But again, I as a physiologist, I work with plants, and the aim is to use it in the breeding program. So that's why I emphasize on the system when we use what we use for screening. Um, Certainly, it has to answer what your question is, but also if your aim is to use it in a breeding program, then does it matter to get all the roots? Because we have been using the 2D measurements. Back in the day when we worked with common bean, we even score. We don't even need the whole exact measurement, but we use the scoring system. So I think it's up to your question.
Oh, there was a problem here. I think I don't know what happened. Sorry about that. Yeah, I think we don't have any further questions here. We would like to thank you all for accepting to present our research here, your research here. Thank you very much. And I think many people were able to learn something new today. And this video will be saved in our YouTube channel. So whoever wants to go back and try to run the protocol again will be great, so they can follow all the steps that was taught today. Yeah. Do you have anything else?
No, I also want to thank you for letting me or us actually do the workshop. I hope that it will help a lot of people.
Yes.
Yeah,
absolutely.
thank you.
Thank you, guys.
Thank you so much.
Yeah.
So shall we introduce the next speaker?
Yeah.
So our next workshop will be next Friday, and Garlene is going to tell us about the details.
Yes.
So instead of Thursday, next week it will be Friday. So
Yeah.
And the Eventbrite is already available for registration. And if maybe you can share the flyer, Garlene?
You're trying to open here?
Yeah, me too. I can maybe just tell the details. It will be hosted by Guillaume Lobet, and the title will be Using Functional Structural Plant Models to Extend Experimental Phenotypic Knowledge, and it will be on November the 18th, which is a Friday. So we all hope to see you there.
All right.
Thank you so much.
So just to recapitulate, so next week it will be Guillaume Lobet. He is both based at Forschungszentrum Jülich and the Catholic University of Louvain-la-Neuve in Belgium. And he's going to be talking about computing modeling for plant phenotyping. And sorry, we cannot share at the minute the flyer, but the Eventbrite is available. And we will be circulating it on Tweet and to your email accounts very briefly today. So thank you for joining us today, and we hope you enjoy the workshop. Bye-bye.
Bye.